So today we talk about population genetics, or at least we begin to talk about population genetics, and this is my favorite example of a major misconception about population genetics, which we'll start talking about today. This is the trait of Brachydactyly. Anybody in here brachydactylous, if you want to share? It's particularly short phalanges. I don't know, my hands are pretty small, but no. So it's a relatively rare human disorder. So we've got Brachydactyly. And I'm going to tell you something today that will probably be shocking about the transmission pattern of this disease if you don't know about it already. So we're going to talk a little bit about this, sort of a case in population genetics. The main topic will be the Hardy-Weinberg equilibrium, which most of you probably didn't hear about in genetics, because I think most of you took genetics from me, and I don't usually cover this in genetics. The question is, what happens to a new mutation when it arises in a population? So hopefully you understand that that's why this is important for evolution. Because evolution is all about novel genetic changes and mutations. The only way variety arises in a species or in a population is through mutation. Some sort of genetic change. Sometimes they're bad, sometimes they're good, but regardless, they make a change. And that change can either be selected for or against. But immediately after that mutation arises, two things can happen. What do you think? What can happen to a new mutation when it shows up in a population for the very first time? Pardon? Okay, so it's either selected. So what happens to the mutation if it's selected for? Yeah, its frequency increases. So more and more individuals in the population start to have that mutation, if it's beneficial. If it's detrimental, then fewer and fewer individuals in the population would have it. So just starting out with a thought experiment. Say you've got a homogeneous population of individuals. So you've got 10 individuals, all of whom have a single genotype, big F over big F. And then you get, say, an 11th person, a new variant or mutation arises in the 11th. So you have 10 individuals that are that genotype. And then a spontaneous mutation happens and you have one individual that's big F over little f. So what's the frequency of little f in the population?
Like 1 out of 22 total alleles?
Exactly. So this is not individuals, this is total alleles. So we have 11 individuals, each individual has 2 alleles. So there are 22 alleles, 22 versions of this gene in the population. So little f is at a frequency of 1 in 22, big F would be 21 out of 22. Sorry, that's a comma. So this is what a population will look like as soon as a new mutation arises. You have one individual that's got the mutation. And then you've already kind of given me a prediction about what might happen if natural selection acts on this little f. In the next generation, what's going to happen? You'll either have the same number, or you'll have more, or you'll have fewer. So it'll be the same frequency, or the frequency will change. So this chapter, population genetics, the concept of population genetics is understanding how allele frequencies change over time and trying to predict what will happen to allele frequencies over time. Okay. So same scenario, big F's and little f's. We have a lot of big F's in the population. We have one new mutation, the little f. just one individual is heterozygous. Say you come back to the same population 10 generations later, and you sample the DNA from the individuals, and there are only little f's. No more big F versions of that gene. What would that suggest to you? So it could mean that it was selected for, that it was better than having the big F version of the gene. So all the big F individuals were eliminated from the population over generations. They had fewer and fewer kids every generation compared to the individuals with big Fs. What if the opposite is true? What if you come back 10 generations later and there are no little f's anymore? We started with 1. You come back 10 generations later, there are no little f's. Okay. So that could mean that it was selected against. Other answers?
That person just didn't reproduce, like the likelihood of it happening is less than the other ones.
Okay, so by chance, maybe, that one individual that had the little f allele didn't reproduce. Didn't find a mate, died before sexual maturity. So there are other explanations, that's the point. We tend to think that if we see a change in allele frequencies, that's because selection is acting, either for or against that allele. But that's not the only way that allele frequencies change in a population. So there's at least one other explanation for changing allele frequencies, and that's how individuals happen to mate with each other, or if they mate at all. Any comments or concerns about this point? It certainly could be the case that being heterozygous is the cause for not having mated, in which case this would count as natural selection. But if it was just chance, as you suggested, that the individual happened not to mate, didn't have anything to do with their genotype, then how mating occurs, predation and so forth, might affect allele frequencies over time. So I'd like to remind you, gently, about one of Mendel's findings. That if you take a pure breeding yellow line of peas, and this is again, all has to do with changing allele frequencies. Capital Y over capital Y, pure breeding yellow line, and a pure breeding green line, which is little y over little y, crossed together makes F1s that are heterozygous. So you have 2 heterozygotes. And then what are the genotypes of these F2Ps? There's a particular ratio. So this is a 3-to-1 phenotype ratio. There are 3 yellow peas to every green pea. And inside that, there's a 1 to 2-to-1 ratio of genotypes. This would be results from a Punnett square. So you've got 1 big Y over big Y individual. What's the genotype of a green pea? It has to be, it's the same as the parent up here. So the green is recessive. So you got 1 homozygote big Y, 1 homozygote little y, and that leaves the 2 in the middle being hets. Big Y over little y. Did the allele frequencies change from generation to generation? What's the frequency of big Y in the P generation? The P0, sorry, these are all P generations. Get it? Come on. Yeah, thanks. Okay. The pity laughter, that's what I go for. How many big Ys, what's the frequency of big Y in the P0 generation? So we got 4 alleles, 2 big Ys, so the big Y is at allele frequency of 0.5. Half of the 4 alleles are big Y. Did that change in the F1 generation? How many big Ys are there? What's the frequency in this generation? 50%. What about the third generation, the F2? So we've got 1, 2, we've got 2 heterozygotes, so that's 3, 4, I'm just tallying. 4 out of 8 alleles are big Y. So the frequency of big Y is still 0.5. So from generation to generation, at least in this cross, the allele frequencies haven't changed. They've stayed the same. And the same number of big Ys and little y's every generation. What's going to happen in the F3 generation, though? If you take all of these, so far you might argue, and you should argue, well, this is Mendel setting up controlled crosses in his garden. The P0 cross, the F1 cross. That's not random mating. So let's assume we take all of the F2 individuals and mate them with each other. Sibling mating. What's going to happen in the F3 generation? That's funny, I thought I had another slide in here. Let's do that in a second. Like this. The mutation's dominant. Mendel would have predicted that after 2 generations, 3/4 of the people in the world would have this trait. It's wrong because of allele frequency, that assumption that 3/4 of individuals after 2 generations of the existence of this mutation would be brachydactylous, have the dominant trait, yellow peas or short fingers, is because how common is this trait? It's really rare. So if the mutation that causes brachydactyly is really rare, then in the entire population you won't see it very often, even though it is dominant. So this is how this argument all got started. This fabulous letter to Science, which is, I don't know, I think it's funny, but then I'm kind of a genetics nerd, so there you go. I'm reluctant to intrude in a discussion concerning matters of which I have no expert knowledge. So this is Hardy writing to Science. I should have expected the very simple point I wish to make to be familiar to biologists, outsider, not a geneticist. However, some remarks of Mr. Udney Ewell, to which Mr. Punnett has called my attention, suggest it may be worth making. Mr. Ewell is reported to have suggested that if brachydactyly is dominant, we would see 3 brachydactylous to 1 normal individual. So this is when this whole argument got started. It's almost the foundation of population genetics right here. So if brachydactyly is dominant, why hasn't it spread across the world, basically? So this is why I'm asking for the F3 generation. If you start with this simple cross, you get an F2 generation like was just suggested, this 3-to-1, yellow to green. What's going to happen when we cross all those F2 siblings together? Does the frequency of the alleles change? Do you have, we just calculated this a couple minutes ago. Big Y in this generation is 0.5 and little y is at an equal frequency. So in those 4 individuals, there are 4 big Ys and 4 little y's. In the F3 generation, if we just mate all of those F2 siblings together, what's going to happen to the big Y and the little y frequencies? The ratios will change, but the frequencies won't. Okay. Let's see what happens. So we already wrote down the F2 genotypes and the allele frequencies. That was earlier on. So if we want to figure out what are the allele frequencies in the next generation, we're going to have to set up a Punnett square and figure out what happens with random mating starting with an F2 population. So let's see. I'll write these again. We've got a relative ratio of 1 homozygote, 2 heterozygotes, and 1 homozygous recessive P. Okay. Allele frequencies are 50-50. Half of the 8 alleles are big Y, half are little y. So if we represent those allele frequencies, the 50% that are big Y occupy a column that's 50% the width. So each of these axes just represents the frequency of gametes, the frequency of alleles. We've got 50% big Y. It's in blue. We've got 50% little y's. We'd make equal-sized columns, equal-width columns. Half of the gametes that are produced by those F2 individuals are going to have big Y's, and half of the gametes are going to have little y's. Whose rule is that? That's Mendel's first or second? First, yeah. Right, exactly. The law of segregation, I heard. The segregation of alleles. That everybody's gametes accurately reflects the 2 alleles that they have. If you're homozygote, big Y over big Y, all of your gametes are going to carry 100%, are going to carry big Y. If you're heterozygote, half of your alleles are big Y, half are little y, and half of your gametes have big Y alleles, and half of your gametes have little y alleles. So between the individual genotype and the gametes they produce, there is no change in allele frequency. So within a generation, going from an actual organism to the gametes it produces, allele frequencies stay the same. That's Mendel's first law. We start out with organisms that are 50-50, half big Y, half little y alleles. We go to gametes that have exactly the same frequency. And we write the exact same thing on the other axis because we're mating the same individuals with each other, siblings with each other. So we have 50% big Y and 50% little y alleles. And what the Punnett square tells us is just the relative frequencies or ratios of those those genotypes. So did allele frequencies change from the F2 to the F3 generation? What's the ratio of big Y's to little y's inside the Punnett square? 50-50. 4 big Y's, 4 little y's. Allele frequencies didn't change. That is a critical piece of knowledge to always keep in mind. Just based on genetic principles, Mendel's laws, and random mating, which is what the Punnett square represents, random combinations of all the gametes being produced in a population coming together at random, you don't expect allele frequencies to change from generation to generation, stay exactly the same. Unless, what are the reasons that genotype frequencies could change in a population? These are the exceptions, and there are a few of them. So what was the first one? Linkage. Linkage keeps allele frequencies of linked genes, so the linked genes stay the same. Allele frequencies don't change. Mutation. So how did the allele frequency in that initial population of 10 big F over big F individuals change when that little f mutation that I talked about at the very start of class arose in the first place? The allele frequency went from 100% big F to, what, 95% big F or something like that. So mutation. mutation is the cause of variation, so any type of mutation will change an allele frequency of whatever gene that mutation happens to be in. So yeah, mutation is one reason you might see allele frequencies change from generation to generation. That would break Mendel's rules. There's something having to do with mating. Yeah, in this case, the specific, and we'll talk about this in greater detail as we go through this chapter, non-random mating, or as was suggested earlier, whether or not that individual mates at all. So non-random mating patterns, Punnett square represents random mating. What would be an example of non-random mating?
Mate selection?
Any sort of mate choice that depends on genetics. So in humans, if women only mated with guys that had beards, and if beards were genetically controlled, the ability to grow a beard that is, then you would see allele frequencies change. Men that had the allele that could grow beards would increase in frequency over time. So random mating, non-random mating affects allele frequencies. What about the suggestion before this one individual that's heterozygous, big F over little f, happens not to mate? What are some reasons why you might expect that individual didn't reproduce?
Maybe because the mutation was detrimental and so they couldn't, they died before they could mate or something.
Okay, so that would be, let's see, how many am I trying to write down? 5. So that'll be the 5th one. The bottom line, thanks a lot, so the reason we're all here, natural selection clearly is a force that changes allele frequency. If you happen not to reproduce or you die because you have that allele, die before reproduction that is, if you happen not to reproduce for any reason, sure, natural selection. You've got a heritable variant that causes a change in phenotype that changes your fitness. That will change the frequency of that allele.
What about genetic drift?
Okay, so genetic drift. Dun dun dun. What the hell is drift? Can I explain it in a reasonable way? We're going to get there. Genetic drift has a lot to do with another factor about whether or not an individual is likely to mate. So why might that individual that was big F over little f not have mated? So physically, oh, what about, so remind me to come back to that. Physical movement. How else can allele frequencies change in a population? You either get mutations arising from scratch, de novo, a new mutation in a new organism. How else can alleles arrive in a population or allele frequencies change? Immigration or emigration. So if new individuals physically move into your population and bring mutations with them, that's basically the same thing as getting a new mutation, number one. It's just that they arose somewhere else and they moved into your population physically. So if individuals that carry alleles emigrate, leave, or immigrate, arrive, that process can change allele frequencies. As far as drift goes, the concept of genetic drift, which I'm not going to introduce in detail now, we'll talk about more, has a lot to do with population size. So how big the population is dictates whether or not you expect allele frequencies to change or not. So what's the issue with, is it good to have a big population size or a small population size? Big. What's wrong with having a small population size? Pardon?
Less variation.
Less variation, that's one thing. But in terms of evolutionary fitness,
Yeah, if you get a deleterious mutation, you have more chance of wiping out your population.
so a deleterious mutation has a bigger chance of wiping out your population. What has to happen for a population to sustain itself? Basic biological urges. It's springtime, reproduction. What's wrong with being an individual in a small population? Inbreeding and not many individuals to choose from. What if you happen not to reproduce because you can't find a mate because your population is so small?
Then you lose that too.
Then you don't reproduce, and that allele that you carry, that new little f, disappears because you didn't happen to mate. So population size has a direct effect on your ability to mate. That's one of the reasons that genetic drift occurs. In a nutshell, for those of you that haven't read the chapter or heard about drift before, the concept of genetic drift is anything having to do with random chance events that cause something like the inability to find a mate. So it's directly related to population size. So random events can change allele frequencies, mostly to do with how big your population is. Big population reduces drift. So large population equals a low amount of genetic drift. Small populations are bad because that enhances genetic drift. So these are the 5 exceptions to the rule that allele frequencies don't change. In other words, these are 5 forces that do change allele frequencies. So how is this useful? Maybe I should start with, is it useful? Here's an example. So let's say we come back to that initial population that had one individual that was heterozygous, big F over little f. We come back and the population looks like this. So what are the new allele frequencies? How many little f's are there? 7. So just count them. 1, 2, 3, 4, 5, 6, 7. I got 8. So 8 little f's. How many individuals are there? 10. So that's 10 times 2, or 20 total alleles. So the frequency of little f is 8 over 20. Then you can just do subtraction. The frequency of big F then must be 12 out of 20. So the frequency of little f has increased since we first saw this new mutation arise. Does that mean that little f is dominant or recessive? If you see an allele frequency change over time, does that tell us anything about dominance?
No.
It turns out no. Can't tell. So changing allele frequencies doesn't say anything about whether or not a mutation causes an effect when it's heterozygous or homozygous. What about if it's beneficial or deleterious? We already talked about that. There are many reasons that an allele frequency can change. Little f's. It doesn't all necessarily require natural selection. It could have to do with population size. There could have been immigration of other individuals that happen to carry the same mutation into the population, and so forth. So that list we just came up with, all of those could explain an allele frequency change. So we can't yet tell, just from this information, just the fact that allele frequencies are changing, what would we have to do to know whether or not an allele was beneficial or detrimental? So we'd have to measure fitness. How do we prove if natural selection is causing an allele frequency change?
You could try to control all of the other covariates.
Exactly. I hope you all heard that. I'll restate it in case you didn't. The way that we tell if natural selection is causing a change in allele frequency is to rule out all of the other possible options. So you try to control those other 4 reasons why allele frequencies could change. So if you're an evolutionary biologist, if you're working in the field or in the laboratory, you try to come up with rational explanations for how you know that the population size is large, that is, genetic drift isn't functioning, that there's random mating, that the effect of mutation is minimal, or basically essentially 0, although mutation always happens, so that's always a caveat that evolutionary biologists are working against. Mutations always happen, but you have to come up with arguments for why you don't think, in the case you're studying, mutations explain changes in allele frequencies. So, did I miss one? You try to argue that your population's randomly mating, has a large population size, no mutation, no immigration or emigration. Which is easy to do in the laboratory. You can grow, that's why a lot of the model organisms for evolution are the ones that have large numbers of offspring that you can easily grow in the laboratory. You control whether or not individuals are leaving or not. It's harder to do out in the field, which is why I have the ultimate in respect for evolutionary biologists that deal with real organisms out in nature. It's a lot harder to prove whether natural selection is working when you're studying a wild population. Okay, so given these allele frequencies, 8 out of 20 little f's and 12 out of 20 big F's, what genotypes would you expect to see if selection isn't working? So if you have a population with these characteristics, the frequency of big F is 12 out of 20. And the frequency of little f is 8 out of 20. Then when you go into the population and you measure genotypes, you collect a bunch of individuals, you take tissue samples from all of them, and you find out their genotypes of this gene. Are they homozygous big F, homozygous little f, or heterozygous? What should you expect to observe? What do we have to do if we're going to determine how many genotypes we expect? How do you calculate that? Pardon? Punnett, yeah, I know. Another Punnett square. So how do you draw a Punnett square that represents not an individual cross, but now we're talking about a whole population? We have a bunch of individuals, hundreds, thousands, but we know the allele frequencies. We've estimated them by sampling a few individuals. So when you go out into the bigger population, how do you set up a Punnett square for this? How many columns do you need? How many alleles are there? 2. There's a big F and a little f. So you're going to have some gametes that carry a big F and some gametes that carry a little f. And if there's random mating, you're talking about gametes from 2 different parents, the 2 axes of the Punnett square. So that's parent 1 gametes. That's parent 2 gametes. So either parent, if you're just looking at this one gene, will either contribute a big F or a little f to their offspring. Now what? There's the square. The length of each of these axes represents 100% of the gametes. So how wide do we make the two columns? The big F and the little f. Let's say the width of this column is 1. 100% of the gametes. How wide is the big F column? It's 12/20ths, or what is that? Reduce it. 0.6. So 60% of the gametes in the population will carry a big F, and 40% will carry a little f. So you can represent that in a Punnett square just by changing the widths of the columns to represent the frequencies of the alleles. You may be more familiar with this, unfortunately, as P and Q, where P is defined to be the frequency of one allele and Q the frequency of the other allele. And then you start having flashbacks to high school or Bio 1 about the Hardy-Weinberg equilibrium formula. That's where we're heading. I'm just trying to derive it in a graphical manner first. So the heights of the two columns are the same. If you've got 60% big F's and 40% little f's, you can draw sort of this weird asymmetrical Punnett square. All this does is represents the frequencies of the two alleles and what you expect when they all come together at random at fertilization. So what's the area of the square where big F's come together with big F's? So this is the largest population. So the areas of these squares in the Punnett square tell you something about the relative numbers of individuals that are going to have those genotypes. What's the area of this square? 0.6 times 0.6. Oh crap. Just move the decimals away and do that. 0.36. What about the least common genotype? What's it going to be? Little f and little f together. 0.16. 0.4 times 0.4. No, wait. Is that right?
Yeah.
Okay. So what's big F times little f? 0.24. And there's two of those. Hopefully, if we've done this correctly, all of those will add up to 1. Yes. Whew. Okay. So we've got 100% of the individuals accounted for. 0.36 plus 0.24 plus 0.24 plus 0.16 is 1. So there you go. Those are the relative frequencies. 0.36 out of 1. So about a third of the individuals should be big F over big F. 0.16, 16% of the individuals should be little f over little f. The rest should be heterozygotes, big F over little f, because those two genotypes are the same genotype. So you add 0.24 and 0.24. About a half of the individuals are going to be hets. In other words, the frequency here is width times height, P times P, frequency of big F times the frequency of big F. The frequency of little f over little f is Q times Q, and then you've got P times Q and P times Q added together, so 2PQ. So those are the relative frequencies of those genotypes. That's the Hardy-Weinberg equilibrium formula. P squared plus 2PQ plus Q squared has to equal 1 because it's a sum of frequencies. And that's the essence of population genetics. What the Hardy-Weinberg equilibrium does for us is tells us if we see a population and we have a measured number of alleles, in this case 2, big F and little f, what genotypes do we expect to see? If we go into the population, we pull out a bunch of individuals, and we see that there are 36% big F over big F and 16% little f over little f, when our observed and our expected values match, then we've declared that the population is in Hardy-Weinberg equilibrium. The frequencies of genotypes we observe are equal to the ones that Mendel would have predicted based on his laws. Genetics are working right, or at least the way we expect. So this is, do take note, in the absence of selection. So together, if you combine that with our list of requirements, how do allele frequencies change? If you can control the top 4 requirements, and the only one that's left is, well, maybe natural selection's happening, maybe that's why allele frequencies are changing, then you use this test, the Hardy-Weinberg equilibrium test, to determine whether or not your observed genotypes in the population are the ones you'd expect. If they're not, when the genotypes you observe do not equal what Hardy and Weinberg would have predicted, that means that natural selection is working. Is causing an allele frequency change. So in red up here, when observed equals expected, that is what you expect under the absence of selection. Conversely, when the observed and the expected values of genotypes don't match, then you can conclude that natural selection is causing a change in allele frequencies. But even then, only if you know that there's no immigration, no emigration, no mutation, large population sizes, and random mating, which is a lot, I know. And very few evolutionary biologists actually succeed in determining all of that. What I ask of you is, when you start thinking about writing term papers, if you're going to write it on evolution, or you're going to write it on the evolution or how a trait arises and spreads through a population, think about addressing all of these points in your paper. If you read a manuscript, did the authors prove that there was no mutation, no drift, large population size, and so forth? Okay, so quick summary right there. So here's one example, just a quick one, then we'll end. Do the rest next class. Different human populations, I want you to figure out, or at least think about, are these differences caused by natural selection or not? Blood group alleles. There's 3 different versions of the blood group antigen. There's big A, big B, and there's little I. And here's 5 different human populations. People have gone into the populations and measured how frequent those 3 alleles are. Why do you care? So we care about blood type because of blood transfusions. So next time you get in an accident, if the blood center is running low, and you wanted to have, let's see, which of these is universal donor? Oh, okay, so thanks. So let's say you're near a crash or you're involved in a crash and you need blood fast. You need to get it from somebody. You don't have time to send it to a lab to get it typed. Which human population, a human of which population do you hope is in the area? And you say, hey, give me your blood. Who's most likely to have a universal donor blood type? The Sioux. Holy crap. The allele frequency of little I is 95.5% in that population. Wow. Or you hope that you're the universal recipient, AB, which is most likely to be someone who's, I don't know, maybe either Japanese or pygmy, that have the lowest levels of the little I. So most likely to have combinations of big A with big B. So if you're in a crash, look for someone who's Sioux to donate blood, and you better hope you're either Japanese or pygmy. Bottom line. But the question is, so think about this, what caused the differences in these allele frequency changes? Natural selection? That is the reason that these 5 populations differ because there are important reasons to have those allele frequencies differ? Or is it immigration, emigration, mutation, chance, small population sizes, genetic drift, etc.? So ponder that, please, and we'll keep talking about population genetics next time.